All right, it's my pleasure to introduce Ryanne Murray. She is a research associate in Gulfport, Mississippi for the Global Center for Aquatic Health and Food Security, serving the Gulf Coast Aquatic Health Program. She has a BS in marine science and is pursuing her master's degree under the direction of Dr. Barbara Kaplan and Dr. Beth Peterman. Looking forward to hearing about your work.
Yes, thank you so much. So today, yes, like you said, I'm Ryanne Murray. I'm a graduate student at Mississippi State through the College of Veterinary Medicine, and I work for the Global Center for Aquatic Health and Food Security. So today I'm going to be talking to you about the effects of age on toxicant levels in tissues of dolphins that's stranded in the Mississippi Sound. So a little bit of background. So dolphins, which might not be fish, but they do eat them. These animals are a great indicator of ocean health. Tursiops truncatus are native to the Mississippi Sound. And like Jill had mentioned, they are the largest population stock in the United States for bottlenose dolphins here in the Mississippi Sound. These animals also have really high site fidelity. So that means that we have a resident population of animals that don't really migrate out of this area. So that also leads them to be exposed to a lot of human activity that happens along these waterways into the Mississippi Sound and the Gulf of Mexico. So some of those things would be contaminants, environmental pollutants such as PCBs, so polychlorinated biphenyls, and PHs, polyaromatic hydrocarbons. So that's what I'm going to be talking to you a lot today about. So these bioaccumulate in the tissues of these animals. And one thing I wanted to mention, just some background, is I will be mentioning some of that offloading of toxins that happens for females. So we tend to see, because those females are able to offload those toxins to their young through either the placenta or the milk, we tend to see females having lower values of PAHs and PCBs than the male dolphins, which we'll talk about a little bit more later. So for the sample selection, we had dolphins that stranded in the Mississippi Sound from 2010 to 2021. During this time period, the Stranding Organization Institute for Marine Mammal Studies that responded to all these animals was also covering some of Alabama in the early 2010s. So we included the animals that were stranded in Alabama that were still within that geographic barrier of the Mississippi Sound. These animals had to have all 4 tissue types, so blubber, muscle, kidney, and liver. And they were code 2 or 3 animals. So code 2 is fresh dead, and code 3 is mildly decomposed. And we wanted to make sure we had very high sample quality for the analysis later on. So that's why that was selected. And that left us with about 138 dolphins during this time period. So those dolphins were then taken back to the Institute for Marine Mammal Studies, and they were necropsied, where we collected those samples, blubber, muscle, kidney, and liver. Then they were sent off for analysis up at Mississippi State Chemical Laboratory up in Starkville, and we analyzed for 24 PAHs and 7 PCBs. So getting into the age classification of these dolphins. So straight length is used to estimate the age of dolphins. This is a helpful tool when you don't have other diagnostic measures to age animals. However, its accuracy can vary across regions. For this study, we chose to break up the age classes from these dolphins into 3 categories because the sexually mature animals are considered subadults by NOAA, which is the organization that oversees them and has a lot of the data collection. But because we wanted to capture that offloading in these pregnant females, we considered some of those, what NOAA would consider as subadults, as adults, to capture that. So any animal that was greater than 200 centimeters was considered an adult. Then we have our subadult category, which is 116 to 199. Those are going to be some of your calves and not sexually mature animals, and then perinates, which is less than 115 centimeters. Those are your young animals about 2 weeks before or after birth. So they're going to be the youngest. So for our results, we found that 9 out of the 24 PAHs were detected, with naphthalene being the most frequently found. All 7 PCBs were consistently detected across this time period. And PCB 153 and naphthalene and blubber I used to compare the toxin levels across these ages because, like I said, naphthalene was the most frequently found. Also, they had the highest levels, so it's a good comparison. So PCB 153, just a little bit of background on this PCB. So there are over, I think there's around 200 PCBs. And again, 153 is very commonly found in these animals. So it's an organic man-made chemical commonly used in the electric industry as a dielectric insulating fluid for transformers and capacitors. It's fat soluble. So compounds that are water soluble are expelled, while chemicals that are fat soluble are not. Because this is fat soluble, PCBs can accumulate in animal tissue like blubber and move up the food chain over time. So PCBs were banned in 1979 due to their negative impacts on the environment and human health concerns. However, they still persist in the marine environment and terrestrial environment. So we're still very interested in the effects of PCBs on these animals and its accumulation in their tissues. So some of those effects are anemia, hypothyroidism, reproductive issues, and immunosuppression. So looking at our results here, we found a distinct group of perinates with lower values. So again, those are your very recently born animals. And then we have subadults and adults. So adult males had higher values than females. And you can see there's a positive correlation. Again, so those females kind of drop off right around where I had mentioned this is what we considered adults, so sexually mature animals. So they're offloading those toxins to their young. And the males aren't able to do that. And we do have some outliers in there. And again, the males were statistically significant. However, the females were not. So it's just very interesting. And again, we see that bioaccumulation over time as these animals grow and age. So next is naphthalene. So naphthalene is a PAH. It's found in insecticides, resins, dye intermediates, synthetic tanning agents, moth repellent, and miscellaneous organic chemicals. So this is mothballs are a big one. People know it for. It's also released in the air from the burning of coal and oil. So unlike PCBs, naphthalene is metabolized a little bit differently. So it varies the metabolism of it varies among species. It can metabolize through the urine, and a small fraction can eliminate in the feces. It's not significantly bioaccumulated, but it is temporarily stored in those fatty tissues. So some of the effects of naphthalene can be ocular issues, hemolytic anemia, damage to the liver, and neurologic damage. So looking at naphthalene, it's different. It's a negative correlation here. And we have that distinct group of perinates again with these higher values. So it's kind of interesting that it's different than the PCBs. And again, the males were statistically significant where the females were not. So again, it does bioaccumulate differently due to that metabolism. So to kind of speculate on this, it's probably the moms are offloading that naphthalene to their young perinates, again, through the milk or the placenta. And because of its metabolism, they're dying and they're not metabolizing that like the adults are able to. So interesting finding. Expected the opposite really for these PHs and PCBs. So I'm going to continue to analyze the rest of the PHs and PCBs, and it'll be interesting to see what we'll find. So it's really important for the long-term surveillance. So we're going to continue to necropsy these animals and take these samples and analyze them for the PHs and PCBs to better understand the effect of the overall health of these animals and how these toxins are affecting them. Also, these animals are eating the Gulf fish that's out there. So how is that? What are the human health implications of this? These are sentinel animals, so really, really good way for us to kind of figure out what's going on with our stocks of fish. So future work, tying it again back into fish. So we're going to analyze fish over the summer, do some necropsies, and pull those same tissue samples that we pulled for the dolphins. So muscle, kidney, liver, and fat. And we're looking at sheep's head, striped mullet, hardhead, sea catfish, Atlantic croaker, menhaden, herring, and kingfish. So those are common prey items for these dolphins out in the Mississippi Sound. So we'll be looking at those over the summer, and so we can determine if diet is the source of this contamination. We're also going to increase our sample size by adding more dolphin tissues from 2023 and 2024. So I would just like to acknowledge IMMS for the stranding work that they do, NOAA for the permitting and funding from the Mississippi Department of Marine Resources. Here are my references. Does anyone have any questions?
Great job, Ryan.
Thank you.
We've got a question in the chat. Their network isn't great, so you may have covered this. What could cause the high levels of PAHs?
Yeah, so again, it's coming from like runoff, right, of things into the Mississippi Sound. So we're trying to kind of figure out, and that work that we're doing with the fish is to kind of determine where that contamination is coming from, whether it's food, whether it's environmental. But there are a lot of factors that could contribute to that.
All right, we got another one here in the chat. After the Deepwater Horizon incident, there were efforts to look at sediments and fish samples in the Gulf of Mexico. And those samples, I believe, are frozen and stored. They are curious if there's any efforts made to look into those historic data.
Yeah, so I think that's something that we're trying to, this is my like thesis work. So I just started this semester, so I haven't dove super deep into a lot of these things. But that's something that we'll be kind of looking at is probably doing some literature review on that, and then going forward with what's happening now, and looking at those fish that we're going to analyze this summer. So.
Great. And I'm about as far away from dolphins as you can get in the United States. But so this might be a dumb question. I'm curious, do these different sort of size categories of dolphins that you looked at commingle, or are there sort of spatial differences that might make them predisposed to come in contact with runoff or effluent from stormwater, wastewater?
That's a good question. It's more kind of the geographic range of the different stocks. So more comparing those, like I said, these animals within the Mississippi Sound have a really high like site fidelity. So they will stay in the area even maybe when there's some issues like Deepwater Horizon when that happened, not migrating, the freshwater incursion, not leaving the area when they are susceptible to those freshwater exposure. So they stay. They're resident animals. We do have transient animals that do pass through the Mississippi Sound, but so their exposure varies. And then there's a lot of work out there that's being done with other populations across the United States of bottlenose dolphins, and looking at the different pHs and PCB levels in those subpopulations as well. So they vary a lot based on where you are in the United States and in the world globally.
Very cool. It's a complicated story. Thanks for working on it and sharing it.
Thank you.